Okay, folks, so we're going to look at Unit D, population genetics today. Just a little review on population. When we look at a population, we're talking about the same species, a certain number of them in a particular area in a particular time. For example, the dog population in Edmonton in 2020 is 100,000. Okay, dog is a species, the number, the area, and the time. All four factors have been provided there. When we talk about gene pool, though, we're talking about, let's pretend these are all the dogs in Edmonton, all the different species you can see there. They're capable of interbreeding and producing viable offspring. That's our definition of a species. However, when we look at all the genes of all those, we've got many, many different alleles, dominant, recessive. Some subspecies might have a slightly different alleles than another set of subspecies, but they're all capable of interbreeding. And these are all the gene pools. Now, if you recall when we talked about skin color, the gene pool frequency of skin color genes, its polygenic trait, would be very different in sub-Saharan Africa than it would be in Sweden or Iceland, for example. Weinberg says, what happens if, what conditions would be needed to keep these allele frequencies the same generation after generation? Well, they're going to stay the same if 5 conditions are met. The population is large, so chance events will not alter. Mates are chosen randomly. There's no mutations. There's no migration. There's no natural selection. So if those 5 conditions are met, the next generation, this is generation 2, will have exactly the same allele frequencies as generation 1 here. And when we add dominant and recessive of one allele, they add up to 1. I want you to watch this video by the Amoeba sisters. It'll help you explain this again. But we know these are assumptions. We know that in nature, there are selection pressures. For example, a snake might be able to eat the small frogs. There are mutations. Populations do migrate. Not all populations are large, and mating is actually non-random. So think about dating. You're certainly just not going to date anybody. You're going to be very picky. That's non-random mating. So we can do the math on this. Frequency of the dominant allele is denoted P. Frequency of the recessive is denoted Q, and they add up to 1. But be very careful here. This is the allele frequencies. What happens though if I take 2 dominant, homozygous dominant? Now we're talking about the genotype frequency when we combine them. The genotype frequency for the homozygous, 2 Qs, and PQ in our Punnett square, there will be 2 boxes of those. So we write it as 2PQ, and that has to add up to 1 as well. Please get this out on page 3 of your notes. If I have 500 moths, 320 in this case out of 500 will be homozygous dominant. That gives me 0.64 as my allele frequency for homozygous dominant, P squared. 160 out of 500 will be heterozygotes at 0.32. And the remainder, because this has to add up to 1, 20 out of 500, 0.04 would be homozygous recessive. But now look at the number of genes in the gene pools. So we've reversed this. There's my genotype frequencies. Let's look at the allele frequencies now. Remember, there's 1,000. If there's 500 moths, There'll be 1,000 in my gene pools of A's, little A's, a few little A's here and there. Okay, there's 1,000 of those. So we multiply this by 2, we get 640 because times 2. But also look, half of these, 160, are going to be dominant and 160 are recessive. No, we don't divide that by 2. Remember, that's 1 times, there's 500 moths, right? 160 of them are going to be heterozygotes. So 160 A alleles, 160 little a. And here we multiply this by 2 and we get 40. We'll add these together, we get 800 out of 1,000, gives me 0.8 as my frequency of big A. And we get 200 out of 1,000, 0.2. Well, now we can put these in a Punnett square. 0.91, 0.16, 0.16, 2 times 8, yeah, and 0.04. So 8 squared, 0.91, 0.32, and I made a mistake there, 0.64, and 0.04. And that's going to add up to our 1. 0.8, 0.2 for allele frequencies, add up to 1. And this just shows the same with mice, 0.49, 0.21, 0.21, 0.09. Okay, so let's do a couple of problems here. Randomly made population of snakes, 1 out of 100 is counted as albino. So my Q squared is 1 out of 100. Determine the theoretical percentage of each genotypes, the genotypes. Okay, well, the theoretical percentage of this is going to be 1%. But how do we now get our P squared? Well, we go Q is a square root of 1 over 100. It's going to give me 0.1. Therefore, P has got to give me 0.9. We simply go P plus Q equals 1. So P is going to be 1 minus 0.1, remember? And we get 0.9. Oh, but I need my genotype frequencies. Well, Q squared was given to us. Q squared is 1%. P squared is 0.9 times 0.9, 0.81. So the remainder is going to be my PQs, okay? And we know that we go 2 PQs, and we're going to get 2 times 0.9 times 0.1, okay? And we will get our answer from there. So we're going to get our 18%, yeah, 18%. 0.18, simply multiply these by 100 to get our answers. 1%, 18%, 81% for P squared. A single pair of alleles codes for one of the genes that controls wing length. The long wing allele is dominant to the short. 40 out of 1,000 have short. Okay? So we're trying to now get heterozygotes from this. So we know, therefore, that Q is going to be the square root of 40 over 1,000. And that's going to work out to, if you do the math on this, it's going to work out to 0.2. Now, I want you, if you're not that good at math, no problem, just play around with it until you get this. Okay, the practice ensures that you're doing it properly. So therefore, we can then say that P is going to be 0.8, right? 0.8 plus 0.2 equals 1. So 2PQ is going to be 2 times 0.8 times 0.2. Okay, and then we're going to multiply this by our 1,000. So 0.16 times 2 is 0.32 times 1,000 is going to give me 320 will be heterozygotes. Okay, I would like you to take a look at the textbook questions. The answer key is on the following slide. But before you do that, spend a bit of time on your workbook, pages 4 to 5, and I'll post the answer key later on this week.